Hello everyone, welcome to Standard Biology. So in today's video, I am going to discuss about confocal microscopy. So before proceeding this video lecture, I want to tell you that I had already made video on fluorescence microscopy. If you don't know what is fluorescence microscopy, go and first watch fluorescence microscopy, as confocal microscopy is a modification of fluorescence microscopy. So it will be easier to understand confocal microscopy if you learn about fluorescence microscope. So let's start. So this is a ray diagram of a confocal microscope. So in fluorescence microscope, we already discussed about fluorescence barrier filter, excitation filter, dichroic mirror. But here also I will explain all the part of a confocal microscope. So here we see this is a specimen, or we can say our sample. Okay, and this is objective. This is dichroic mirror. Dichroic mirror have a special kind of property, reflect one kind of ray and transmit another kind of ray. Excitation filter, which allow only a specific kind or specific wavelength of light, and barrier filter, which allow only emission light. Okay, and this is the structure, that is pinhole aperture. Pinhole aperture is not present in fluorescence microscope. So what is the function of pinhole aperture? Pinhole aperture eliminate out of focus light rays, and it allow only in focus light rays. So you can see the out of focus light ray, which is lighter red in color. Okay. So if we compare with the fluorescence microscope, as compared to fluorescence microscope, in case of confocal microscope, excitation source is laser light. So here laser source is present, from where the laser light is present, and it will pass from the pinhole aperture and then from excitation filter, and these excited light when fall on the dichroic mirror, these are reflected by the dichroic mirror, and these fall on the sample, on the specimen. Now these excitation light rays will hit the sample or the fluorophore present in the sample, and then emission takes place. Then this emission light will again fall on the dichroic mirror, and it will transmitted from the dichroic mirror. Then it will fall on the fluorescence barrier filter. The fluorescence barrier filter, which allow only emission light, but it is not able to remove or eliminate out of focus light rays. Now when these light rays fall on the pinhole aperture will remove out of focus light rays and only allow to pass in focus light rays. Okay, now these in focus light rays will fall on the detector. Okay, so this is the ray path which is present in a confocal microscope. So now we will understand that what is the difference and what are the modifications in the fluorescence and confocal microscope. So now we will discuss about that what are the modification done in the fluorescence microscope to make it confocal microscope. So firstly, the pinhole aperture will increase the resolution of image, right? Because it will eliminate out of focus light rays. So by eliminating out of focus light rays, resolution of an image will be better. Okay. Optical microscope creates sharp image that would otherwise appear blurred when viewed with a conventional microscope. So in confocal microscope, why sharp image is produced? Because it is able to eliminate the out of focus light rays. But this does not happen in fluorescence microscope and in conventional microscope. That's why blurred and low resolution images will produce in case of conventional microscope and fluorescence microscope. Okay, and confocal is defined as having the same focus. So confocal itself, it's in its name it tells which a microscope which having the same focus. What is the meaning of same focus here? That is, focus of image and focus of object will be same. That's why the name is given confocal to this kind of microscopy. Okay, so confocal microscope is able to filter out of focus light from above and below the point of focus in the object, right? That's why it will produce high resolution images. Normally, when an object is imaged in the fluorescence microscope, the signal produced is from the full thickness of the specimen, which does not allow most of it to be focused to the observer. The same thing is given here, which I already told you, that fluorescence microscope is not able to produce high resolution images, that is due to it's not able to illuminate out of focus light rays. So same thing is repeated again and again, and light from above and below the plane of focus of the object is eliminated from the final image. The confocal microscope uses a laser beam to illuminate a specimen. The basic difference between confocal microscope and fluorescence microscope is we use different light source. In case of confocal microscope, we use laser excitation source. In case of fluorescence microscope, we use lamp which produce UV or visible light, right? And also another modification is pinhole aperture. Pinhole aperture which helps in eliminate out of focus light rays, which increase the better resolution. Third, which is very important feature of confocal microscope. In confocal microscope, by changing the focus, by changing the focal length of laser light, we can see different layers of a sample. So if we use the whole mount means if we use a whole pollen grain without its sectioning or whole ovary. So by changing the focal length of laser, we can see the internal as well as the outer structure and different layers of internal structures. These are pollen grain optical sections basically. So here we can see that when we change focal length of a of laser, then uh, first image, second image. So as we change focal length of laser, we will observe different structures of pollen grain. So in this way, optical section will be created uh, by changing of focal length of laser, right? So this is whole amount we used. So specific focal length, we are able to see internal structure, the different layers of internal structure and surface layer. Okay, so in case of fluorescence microscope, we are not able to use whole mount. As we use whole amount, we are only able to see the surface structure. That's why sectioning is done in if we want to see the internal structure in case of fluorescence microscope. But this is another advantage of confocal microscope, that we can see whole internal structure and outer structure of a specimen. So I hope this video is helpful for you understanding the confocal microscopy. So thank you so much guys. If you like this video, then hit the like button and subscribe this channel.